So before, I'd like to have a very quick introduction to both speakers first. So, so the first speaker is Dr. Paul Sternberg. He is from Caltech. So Paul got his bachelor degree from Hampshire College in Amherst, Massachusetts, PhD in biology from MIT, did a postdoc at UCSF. He joined the Caltech in 1984 and has been working there since then. So it's quite a long time. Currently, he is the Brand Professor at Caltech. So his research focuses on C. elegans. I guess everything about this little animal using all kinds of approaches, covering all aspects in biology from experimental biology to computational to bioinformatics. So he is one of IDG's external scientific board members. Currently, he is one of the lead PIs of the Alliance of Genome Resources project. His talk today is about curating, integrating, presenting information about the model organism at the Alliance of Genome Resources. So go ahead, Paul. Thank you. Yeah, so everybody, if you have any question, please use Q&A or chat. So.
Okay, it's great. Thanks you for the invitation and participating, and it's a great pleasure to be on the virtual podium with Lincoln Stein, long-term collaborator on worm-based and other projects. So I want to summarize some of the what I hope are relevant aspects of the Alliance of Genome Resources. So the goal of this project is data harmonization to allow common infrastructure for better resource utilization, common views of research organism data, not model organism data, and comparative analyses. And of course, the common infrastructure, one of the things it does is allows economies of scale as well as rapid propagations of advances across all projects. Because if you can normalize the software, then you can implement it once and it'll apply very broadly. Okay, so the complexity here, very briefly, is we started with trying to integrate and coordinate 7 projects that are at 12 sites listed here. So the Gene Ontology Consortium, central, yeast, mouse, worm, fly, fish, and rat databases. And in addition, it's growing because, for example, the frog database is starting to come on board. Okay, so, well, what does that mean? Okay, what do these knowledge bases do? Well, they get data in, they store, integrate, and compute it, and they put the knowledge out, or they add value. So the kinds of data coming in are genome sequences, ontology, sequence features, so anything tied to sequence, special data sets from that organism. From the literature, there's just a host of rich information, including gene expression, phenotypes, variants, gene ontology annotations. But even any of those are also really very complicated. So gene expression can be based on reporters, antibodies, experiments, in situ hybridization of various flavors, including seq-fish and et cetera. RNA-seq and single-cell RNA-seq, old microarray data is still relevant, qPCR, et cetera, experiments. So that's very complicated. And it's done a little differently in each organism, each system. The knowledge out is also complicated. So there's gene reports, I'll describe a little. There's genome browsers, anything tied to the sequence. So like most of us are using JBrowse, developed by Lincoln Stein's group. Data analysis, data integration, and reporting platforms like Intermine, simple, very well-known things like BLAST. This also, the data is ingested by other resources for their integration and display. Okay, so a few years ago, basically we had this siloed approach where there's data coming into worm-based, fly-based, et cetera, and then information coming out. What's currently happening is, and this is some of it's common, shown in gray, and specific to those organisms. So what's happening now is kind of even more duplication where information is coming into the Alliance Central, and information is flowing from some of the information from the individual model organism databases is coming to Alliance, and that's displayed. So that's really complicated and sort of a worst case transition state. Eventually, what will happen is the model organism database projects will be getting in specialized data sets through curation and literature curation. That will be stored as much as possible centrally, and then it'll come out and get ingested in as easy a way as possible. So that's the challenge. Organizationally, it's very complicated. I won't go through this, but the individual model organism databases we envision are knowledge centers. There's also other ones, from around the world. That information gets ingested and computed and then released in various ways to different communities. And that's both downloadable from the cloud through APIs and also through a set of portals that the Alliance is producing. Some of it's still now going back to the model organism databases for the display on their portals. Okay, the architecture, which is I guess one of the things I was asked to talk about, that we're developing now is API based. So to the extent that we can, we're trying to centralize a persistent data store that will allow computation, similar computation of harmonized data. But that's going to be complicated and have individual APIs in that corresponding to a different schema. There'll still be satellite databases that have either large sets of data that don't need to be integrated as much, like variants and from the millions of variants, and information, for example, about all the orthology calls, literature, etc. And these APIs are going to be managed through an API gateway that will keep track of and keep everything clean, and that will allow information coming in from curation and including large data sets. For example, that genome sequences from various organisms and the gene models, and then the output would be through the user interfaces as well as downloads, and the APIs, of course, could be used directly for anybody else in the world. So this architecture is modular, scalable, and flexible, and it's amusingly, or not surprisingly, amusingly, this is what Lincoln's group set up for Wormbase, say 10 years ago, was when we modernized the infrastructure, was to make it as flexible as possible, so that to the user, they don't necessarily know where the information is coming from. It could be coming from any of a number of databases to a number of processes. So you're trying to keep it very clean and simple for users. You can separate out the ability to curate information. You can develop that, because those can be very complicated systems that use text mining and machine learning and other AI to help the humans, the experts, match information to ontology so that it can be computable, because that's the goal of curation, right? So that's so what we're trying to do is make this flexible enough to evolve the system, to make the system work, and to evolve in the future. And of course, it's scalable. We have hundreds of thousands of users, we think, although we don't know because each site has a siloed approach, but there's a lot of users with different requirements. Okay, so I'll talk a little about data harmonization and just sort of how the common infrastructure is already helping us. Okay, so a key aspect of comparative genomics is orthology. So before this project, we had each group had their own way of calling orthologies, that is, evolutionarily related proteins and genes. So now with a common set based on a subset of what Flybase started doing, of a very simple combiner algorithm, you can then get, say, for example, for human MAP kinase 1, you get the ortholog calls, in this case 1-to-1, from the model organisms covered, and you get some information about how many algorithms call them. Is this the best reverse call, which tells you, the 1-to-1 or 1-to-many. Using that information, you can then take harmonized data and do comparative, have a comparative view. So, for example, here's the various BMP orthologs in the different models. And this is what disease associations have been made in the different projects using a simplified ontology, a slimmed ontology, and this kind of ribbon diagram. If you actually clicked on one of these boxes, you would get all the details of the annotations that fit up, for example, in the integumentary system diseases for BMP4, for example. Similarly, you can look at gene expression, and here's major tissues. It is very vertebrate-centric, and we'll work on more clade-specific views. But you can see the BMP4 expression is in, alimentary system, et cetera, and nervous system. And you can see where there are annotations and where there aren't. The density of the annotations is shown by the color, and you can get all kinds of details. But before this, having common orthology and harmonized data in one place was going to be very difficult or impossible to do this. Another benefit is making it really easy for people to get data. So this is, in part, is downloads. Here are disease association files that are associating genes to disease ontology terms for different organisms, or you can get the whole set. Or you can do that by APIs at this Swagger UI site. Okay, so let me give an example of what you do. Let's say you take a random ion channel. Okay, I really was random. I looked it up. Pharos, the illumination graph showed it was not highly illuminated. So what would we get out of the Alliance? So putting that in the search box, we get the human gene. This is the summary, and you can see there's a whole bunch of other aspects, some of which are there, not as much for human because we're really dealing primarily with model organism data. But you can see sort of the overview of this, the description that our RGD, the RAP database uses, which is actually from RefSeq 2011, showing how fast that field's moving. If we go to the orthology table, you can get a set of orthologs. There's 4 Drosophila orthologs, 3 C. elegans orthologs. I just picked sand, sounded interesting because it had a specific name. It probably had something to do with sandwich, right? It's a fly gene, or the beach. So we click on that, and now we get a gene report from fly-based, similar. It's the same format as human, worm, fly, zebrafish. So you don't have to be an expert in all these knowledge bases to get the information. And in a click, you now have a summary of what's known in Drosophila, and it actually has to do with sleep-wake cycle. So it's actually called Sandman, which is an old story about kids falling to sleep. I think it's a Western European origin. Okay, and then you can drill down on information in flies. And here's, for example, alleles and variants. There's one allele. And then you can look at the phenotypes. There's actually 4 phenotypes associated with them from 2 papers, so you can get to the papers. So that's just an example of how you'd go from any human gene or any gene in any organism to the orthologs. If you're interested in the sand gene, you would also be able to start with that and figure out what the human ortholog is or the worm ortholog. In practice, so as a C. elegans researcher, I look at a lot of information. I always filter every gene through worms. So what I do now is I start with the Alliance. If I hear a talk about a Drosophila or a human gene, I type it in the Alliance portal. I figure out what the worm gene is. I can get a summary of what it is, what information I need to know. If I want to actually do an experiment, I go to Wormbase because it has more information now, right? But for the sort of high-level summaries, this works much faster. Okay, let's see what time it is. Right. Okay. Yeah. So that's the harmonization helps. We're still harmonizing away. If I have time at the end, I'll talk about the sort of long tail of information that needs to be harmonized. But we're starting with what's the sort of the high value, what's relatively easy to do, and now we're digging into the hard parts. It turns out, for example, that every group, the way they define genotype was a little different. Even how they define transgenic alleles is different because there's some of it's history, some of it's arbitrary, and some of it has to do with the biology and molecular genetics of that organism. But the idea on the harmonization is to try to get it to as few possibilities, and not 7 or 100, 2 or 3, right? Because there are definitely always different flavors. Okay, so the common infrastructure allows things like data store, working on ID minting, which is we're working on a common system now. Ingest pipelines, we started to continually move over pipelines that are done redundantly from different sources. So every group was taking in all the sequence information to do comparisons. So that we get rid of that. Literature curation is a very complicated, highly evolved systems from the groups that have been working for 20 or so years, and we're trying to rebuild even a better system. As we already talked about APIs and standalone tools are more easily maintained if it's done centrally, because you need an expert who knows JBrowse and how to tweak it. Okay. One thing we're doing on the infrastructure is we're exploring, and I think it looks quite promising now, to use this LinkML, which is a common data modeling language written in YAML. So it's readable by humans and computers, and both biocurators and software developers can use it to communicate via common language. And it's not linked to a specific implementation, but it can be translated automatically, or worst case, with some scripting to APIs, curation interfaces, and database schemas. So that's going to be the common language. And we found at Wormbase over the years that using a common modeling language allowed the biologists to think as much as possible as like the computer scientists who are doing the software development. Okay, I'll just give another example of rapid propagation. Okay, so one of them is we have structured data, structured information and knowledge, and we like to turn that from the computer-friendly versions of that, tables of terms, into something human readable. So these concise descriptions or summaries of a gene function were done in various ways, sometimes automatically in a kludgy way, and sometimes manually, but that of course doesn't scale over, say, 20,000 genes and per organism, with lots of information coming in. So we have a way of using the ontologies, what information we have, and then output gene descriptions. So what does that mean? Okay, here's a manually, handwritten one from C. elegans, which was out of date for this gene, some dual specificity protein kinase, and it's expressed in various places. It's orthology, and it's localized in the nucleus. The automated description is up to date and includes disease, interacting proteins, etc. Okay, how do we do that? Well, the key is, if you think about it, that if you say this gene is involved in 50 diseases, you don't want to read that. That not becomes readable. That's better to use on a table, or it's expressed in 30 different places. So you want a high-level view. So the way we do that is that we take the ontology. This could be gene expression or gene expression by anatomy or gene ontology, various gene ontology terms or diseases, and you find common nodes. So here's these would be where there's annotations, a lot of them. And so these two have a common ancestor, so parent, so you use that. These three have a common parent, you use that. These have a separate, that's it, that's the annotation. So we would refer to those three terms. You also have some limit on how high you go, because you don't want to say this protein is expressed in a cell, it has a biological function, and it has a molecular function, and it's expressed in tissue anatomy, right? That's useless. So you have to kind of balance out where you get the information. So we've done that iteratively to sort of fine tune the algorithm, and that works. And what we've gotten out of that is, for example, gene descriptions that work for the zebrafish database never had these, now they have them. What's interesting about these is that these summarize concisely in very easy way information you have in your knowledge base. So it's a really good quality check, because if there's something wrong, it's not due in general to the algorithm. It's not the algorithm, it's the underlying data. We found that the easier it is for people to see the data, the more errors, mistakes, weird stuff we see. Okay, I just want to finish with one other thing we're doing on the literature creations front is we're allowing us to take advantage of the evolution of literature creation. So 20 years ago, there was expert curation, where you have the biologists looking at papers, putting information in to matching it to ontology terms. Then you have text mining with curator review, expert review. That's work. There's our Texpressa system that we developed has been applied in cases for that. The Saccharomyces pombe database has this tool Canto, which leads authors through curation, and this has been expanded into other model organism databases. But the penetration hasn't been great. So that's it works and it needs to be applied more broadly. The current state of the art is actually the software looks at the paper, figures out what's in it, presents it back to the author to review that. Of course, that's, if you think about it, that's really a backwards way to do things, because the author actually knows that. So what we'd like to do in the future, and we have this micropublication biology project, which is developing this concept, is have the author make the information computable as part of the publication. So we're exploring that in these very small articles. Okay, so those are examples of how you have this set of, these are 5 different types of curation. They're not all applied broadly. So by having a common system, we can then use that and get them really efficient. And the effort put into, for example, getting a good text mining system is only worth it if it's applied across scale. A lot of people involved in this project. And I'll just say that the main thing we want to try to do is say, look, 2 ways are good, 7 ways are bad. And where our goal is to harmonize what we can do in the next couple of years, and then we'll modularize what we cannot so that we don't lose any service. Yeah, I think I'll stop there. Thank you for your attention. And we're not going to do questions till later.
I think we still have several minutes. Probably we can have you probably answer 1 or 2 questions. Okay. So yeah, right now we have one in the chat. Paul, can you see it? Do you need me to read this for you?
I can, if I end my show and go back here, I can see chat.
Yeah, from Jeremy.
Is there a recommended measure of relevance? I guess the relevance would be, is the information you get from that useful and actionable? I think Lincoln's going to talk maybe about data integration. I don't know how he's going to do that, but I know if you ask what kind of predictions can you make from a type of information about from one organism to another, you can actually measure that and ask what's the value. And when we did that, I don't know, 10 years ago or so, there was you could get a pretty good quantitative measure of that type of data. Okay, but in any specific case, like, is this interacting gene going to be relevant in the human disease? You got to do the experiment, right? But you can use evolution to test, say, if these proteins interact in Drosophila flies and yeast, and we test them in fish, then they're going to probably interact in human. So that answers the question.
Okay.
So predictive value, I guess, is what I would say, Jeremy.
Okay, I think we will move to Lincoln, and if you have any more questions, just put it into the chat box or put it into Q&A box. So whatever you like to do, and hopefully we have some time left.
Yeah, and if anybody has ones later on, or is more interested in the Alliance, etc., please feel free to email me.
Okay, great. Thank you.
All right, thank you.
The second speaker is Lincoln Stein. So here's a quick introduction for Lincoln. So Lincoln got his bachelor degree from Johns Hopkins University, MD and PhD from Harvard in 1989. So after that, he did not go to be a doctor and started his career in bioinformatics. So first he served as the director of informatics at MIT Genome Center for 5 years, and then worked at the Cold Spring Harbor Laboratory for another 6 years. So after that, he moved to OICR in Toronto. Currently, he is the head of adaptive oncology at OICR. Among many achievements in bioinformatics, he led the development of the first physical clone map of the human genome and run multiple large-scale data centers for projects like the SNAPE Consortium, the HyperMap Consortium, the Model Encode project, and right now the ICGC project. He's also the lead PI of the Reactome project. Together with Paul, he also leads the creation and development of Wormbase. So his current major focus is on cancer biology and all informatics needs related to cancer data analysis and visualization. So today he will talk about interesting title called Guilt by Association, a few tricks for finding and characterizing cancer-associated genes. Here, Lincoln.
Thank you very much, Guanming, and I'm very honored to be presenting here. As I was working on this talk, I felt that it was, I decided to broaden it a little bit and make it more generic. So I changed, so instead of talking just about cancer-associated genes, I'm going to be talking about techniques for guessing the function of understudied genes in general. This talk was actually adapted from a Canadian bioinformatics workshop on high-throughput biology from sequence and networks given by Quade Morris. I borrowed several of his slides with permission. So bioinformatics has a bit of a reputation for being a little inbred and circular, with a lot of activity around refining and making incremental advances on other people's tools, and only occasionally spinning out a really useful tool that the rest of the research community can use. Fortunately, the problem of identifying putative functions of genes based on their interactions is not one of those cases where there really are very useful tools available that are accessible by non-specialists, general laboratory people. So the general challenges here is that you've done some sort of empirical experiment and you've identified a gene, for example, a suppressor enhancer CRISPR screen, and that gene is functional in your system, but you don't actually know what its context is and what it's doing biologically. And when you go to the usual suspects, such as the UCSC genome browser, you don't get anything more informative than its gene structure. And if you go to the big databases of genes, such as Entrez or Gene Cards, it doesn't tell you very much. In this example, G protein coupled receptor, specific function not determined. How do I know what to do, what experiments to do next? And so in this case, the investment that the research community has made over the past decade in high throughput gene interaction studies really does pay off. There have been many, many dollars poured into making genetic interaction data maps, protein domain similarity networks, protein interaction data sets, high throughput expression data, and curated efforts such as the gene ontology and pathway and curated pathway databases. And these can really help even when the exact function of a gene is not known. Generally, we're talking here about a class of networks, gene networks called functional interaction networks. where you have a series of genes, it may be the whole genome or just a portion of the genome, and you're describing pairwise gene-gene or protein-protein interactions, some of which are directly measured, such as protein interaction networks that are coming from co-immunoprecipitation and proteomics experiments or yeast-2 hybrid experiments. Some of them can be genetic interaction networks where you're looking at suppressor-enhancer screens or the results of knockdowns. And others are inferred interactions. A good example of this is co-expression, are networks from gene expression data, from microarrays or RNA-seq, where you need to do some analysis to identify genes which are expressed under the same conditions more frequently than you would expect by chance, and therefore are under common regulation and may be doing similar things. You can also build networks by combining multiple data sources. For example, genetic interaction plus protein interaction plus co-expression plus gene ontology expression to create functional interaction networks. One of the best of these actually was created by our host Guanming Wu, who created a functional interaction network using machine learning on curated data to weed out high quality interactions from high throughput data. But there are a large number of these inferred interaction networks that have been published over the last decade. An example of a typical function interaction network is to take a microarray data. This is an old data set from 1998, Michael Eisen's original microarray set. You're surveying a series of genes under multiple conditions. They could be different tissues, they could be different stresses or drug treatments or whatever. And then you cluster them by co-expression modules, by co-expression, in order to identify modules of genes which are under the same, which are being expressed at the same time in the same place and are presumably functionally related to each other. So how do you take the, and there are in fact literally thousands of these networks. So how do you leverage all this data to identify the function of a gene? Well, there are basically three general steps. If you have a single protein of interest, you try to identify other better known genes that are related to your protein in some way or another. Maybe they're expressed at the same time in the same cells, they co-locate to the same cellular compartment, they share similar functional domains, such as binding or biochemical domains, they physically or genetically interact, or they share similar known or predicted biochemical activities. Or if you have a set of proteins of interest, you can go further. Now you can ask, is there something, is this set of genes or proteins unusually overrepresented in a particular biological pathway or process in a way that you wouldn't expect to happen by chance? So for example, if you were looking at genes from the mitochondria, you would find an overrepresentation in genes having to do with oxidative metabolism. You can then apply a gene recommender to expand the set using the birds of a feather flock together principle, and then follow that by a gene set enrichment analysis to increase your power to detect these patterns of enrichment. And then finally, once you have a set of genes that are functionally related to each other, and you know what pathways, candidate pathways they're enriched in, you can do pathway visualization following either of these steps in order to dig down and start to make hypotheses about particular interactions and functions that are plausible for your gene of interest. So graphically, the two ways they work is if I have a single gene, I'm interested in asking, what does my gene do? I take as input the network and gene expression profile data, my query list, in this case, a single gene, give it to a gene recommender, which will give me a set of genes which are seem to be related by integrating across these various networks to give you a cluster of related genes. And then you can pass this to a gene enrichment analysis to identify functions which are overrepresented in this cluster that start to give you the substrates on which to make a hypothesis. So for example, it's associated in this case with the yeast budding and asymmetric growth. If you have a set of genes, a query list of a dozen genes here, you can pass that to a gene recommender, and that will expand the list by finding additional genes that connect these, that are connected in a statistically significant fashion. And then you pass that to a gene enrichment algorithm. And then again, it'll give you the same pathways and perhaps some more that came through these indirect locations of the genes that were added by the gene recommender. There are two main algorithms for predicting gene function by finding guilt by association. One is a more conservative way, which takes your list of positive genes and then finds things that directly interact with them. And then a more, does this have an annotation? Yes, okay, this has animation on it. Then a more expansive way of doing this is to allow some indirect associations using an algorithm called label propagation, where in a disciplined way, it finds additional indirect associations, including to genes which do not directly interact with either the genes that you provided or the first set of interactions. It allows it to spread further. So label propagation will give you a larger list of genes, but it may be more speculative. This is a more direct interaction, is a more conservative approach. So the two most widely used tools that I recommend for this type of guilt by association analysis are String and GeneMania. String is the leading direct interaction method. It's highly focused on protein-protein interactions, and its big advantage is it has a lot of connections to structural databases, so that if you find an association, you can immediately go between your gene and another gene, you can immediately compare their structures or look for functional domains in the structures. It makes use of 8 pre-computed functional interaction networks that have been well curated. It has a large organism coverage. It covers hundreds of organisms, and it weights the interactions by an algorithm that calculates the probability of a true functional interaction. And its authors are Pierre Bork, Lars Juul Jensen, and Christian van Meeren. GeneMania is the leading label propagation method. It focuses on genes, and so it will take things like genetic interactions into account. It has many more networks. In fact, it integrates over 2,000 networks of various sorts: gene level, protein level, microRNA level, and curated. But it covers a fewer number of organisms, just human and 8 model organisms. It depends heavily on genetic interactions, shared phenotypic information, and it also has takes chemical interactions into account, which is of interest if you're working on druggable proteins. And it has built-in enrichment analysis built into it. Authors are Gary Bader at University of Toronto and Quade Morris at Memorial Sloan Kettering. So let me take you through an example of using these tools to predict the function of a gene. And to do this, I went to the IDG page on understudied proteins, and I basically just picked out one of their understudied proteins at random. I picked out NEC1. I've never heard of this before. I went to gene cards, and it's actually associated with some structural childhood diseases, short rib thoracic dysplasia 6. It has some gene ontology associations, but not very much is known about it. It's a kinase. So let's use string to find out what NEC1 might do. So very simple interface. You go to stringdb.org, type in the name of the protein, NEC1, do a search. Okay, this is showing you a little expanded magnification of that. And a couple seconds later, it comes up with a protein interaction network. NEC1 is at the center, and it's surrounded by an aura of genes it interacts with, and it shows the interactions between that. It gives you some information on the significance of this network. It's got a p-value of less than 0.05, so it may be usable. And at the bottom, it does a gene overrepresentation analysis shown here, and it's actually very consistent. It's picking up Go terms having to do with the cilium assembly, interciliary transport, microtubule-based processes. These all have a low false discovery rate. So it's picked out a function. This may have something to do with microtubules or cilium or flagellum or something similar to that. And that actually is consistent with a structural birth defect, because ciliary motility defects are associated with a number of structural birth defects. That's great. Let's see if we can get some more information. Let's try Gene Mania. So Gene Mania has another very similar, very simple user interface. You type in the name of a gene or a list of genes in the search box, and it comes out with a network. Again, NEC1 is here. Notice that this is a larger network than String came up with, because it uses other types of information. It has physical interaction networks, co-expression networks, predicted interaction networks, co-localization pathway, and various types of interactions. Now I can mouse over these various interactors, get information on this. You can actually drag the nodes around and rearrange it in various ways. What I'm interested in doing, though, is seeing what these other genes, what processes may be involved. So for Gene Mania, you actually have to select which genes to include in the overrepresentation analysis. So first I selected KIF3A, and I added that to the list of genes I want to do an overexpression analysis on. And then I did a gene set enrichment, and sure enough, I'm coming up with this same thing set that String had: microtubule-based movement, spindle microtubule, microtubule-based transport, and so on, with decent false discovery rates. But I want to go further. I saw P53 there, which as a cancer biologist is very interesting to me. So I added that to the list. And now I get, in addition to vesicle, to the microtubules, I'm also getting cell cycle, checkpoint, DNA recombination, and recombinational repair, and a number of interesting repair genes like MDM2 and BRCA1 are also coming up. All right. Other things you could do are to, you can tune the Gene Mania algorithm by adjusting which types of interactions will go into making its modules. And some of them are turned off by default. If you are interested in transcriptional factor networks, you could turn these, or microRNAs, you could turn these on. Drug interactions, all very useful. In this case, I did not, I just left the defaults. The other thing you can do is to change the algorithm by increasing the number of genes in a cluster and changing the network weighting. One of the more interesting ones is to ask it to weight by gene ontology. If I'm particularly interested in cellular localization, I could have it weight by gene ontology, cellular localization, and then it would put genes that share similar Go ontology terms together. Okay, but after iterating on this in a similar way for a while, I added KIF-AP3 as well. I actually came up with 3 main enriched functions from Gene Mania. microtubule assembly and function, cell cycle checkpoints, and double-stranded DNA repair, which obviously these last two frequently go together. Did we get it right? Well, I go into the literature to search for these things. In fact, for microtubule assembly and function, yes, there's a paper published in 2008. It says that NEC1 kinase is involved in cilium formation. Gene Mania said this was involved in cell cycle checkpoints. Yes, indeed. Searching for NEC1 and checkpoint came up with a bunch of articles, including this one. And finally, is it involved in double-stranded break repair? Yes, it appears to do that as well. Homologous recombination. I searched for double-stranded break repair and homologous recombination, and that came up as NEC1. And in fact, one of the genes that NEC1 is said to regulate appeared in some iterations of the Gene Mania network that I made. So it did pretty well in this case. You might now want to go and dig into this a little bit more to generate hypotheses. Bowstring and Gene Mania allow you to download the data. In Gene Mania, you do it by clicking on the floppy disk icon and clicking genes data. That gives you a list. And then you can take that list and put it into your favorite visualization system. My favorite visualization system is Reactome, that both Guanming and I are PIs for. We go to the analysis tools section, cut and paste the list of genes from Gene Mania in this case, and click continue. That does an overrepresentation analysis, which gives us exactly the same list of pathways that we found earlier. And then you can select one of these and dig into it. You can get a pathway diagram in which it's highlighting where in the pathway each of the genes in the set is appearing. And for complexes, it'll show you the proportion of the complex that's occupied by one of these genes. You can then dig into this even further and explore it and use this to design your next great experiment. So in summary, guilt by association is a really useful technique for getting clues to the function of an understudied gene. It's a good hypothesis generator. String and Gene Mania are two of the most accessible online tools. String is more conservative. It uses direct physical interactions. Gene Mania uses direct and indirect interactions across many different interaction types. It may give you more hypotheses. It may also give you some more false leads, so you need to be careful. Enhancements to this is the example I showed is using a single query gene only, and there's limited information there. Both the algorithms work much better when you give it a set of multiple genes that are functionally related that came out of an experiment. And there are lots and lots of tunable parameters that I didn't have time to show you, but I urge you to explore it. It's really, these are really fun tools to use. And I want to thank everybody for your attention, and I'm happy to take questions.
Thank you, Lincoln. So if you guys have any questions, yeah, please post it to the chat box or QA box, and can ask questions for both Lincoln and Paul. So.
Okay, there's a question, I think it's for me. It's from Natarajan. Using literature information to validate functional associations, especially if the literature is used in network generation. So that is points at circularity. If the literature was used to turn on to generate that association, for example, for a curated pathway database, then it doesn't make much sense to use the literature to validate it. Absolutely, I agree completely. What you can do with these, what you can do, however, is you can turn off. You can turn on in Gene Mania, which does use literature annotations. You can turn on and off networks that were based on the literature. So you could turn off the Go annotations entirely. You can turn off pathways, and then it's just using literature agnostic, high throughput data. But again, as I say, these are not, the networks that you get out of these tools are not definitive publishable results. These are good for pointing you in the right direction, helping you make a hypothesis. They need empiric validation. And going back to the literature is useful for validating that the tools are not completely giving you a red herring, but is not definitive either.
Okay.
I'd also say that in response to that is that it's also a way of finding the papers. So a lot of times you would find information, you would never find that paper or remember it long enough to integrate with other information. So, I also have to worry about circularity, but I think that, you can, if you see that paper, you go, I should have known that, but actually you would really have trouble finding it. So these networks can be really useful that way.
Lincoln, I have a question. So in network, right now, people always focus on like protein-protein interaction or some really pure protein interaction network. The network nodes are always proteins or genes. Do you think that will it help, like if we integrate multiple different types of information, for example, like disease, then create a kind of heterogeneous network because genes, protein, let's say disease phenotype or literature reference, so-called knowledge graph. With that will increase the kind of probably more likely reduce false positive rates because usually probably biologists probably just have too many results coming out from this analysis.
Well, I think there's two things you can do. So one is one is the widely recognized problem that a lot of the functional interaction networks and gene co-expression networks don't take the cell type into account because they were done on whole tissues, right, or whole bodies. And in fact, it doesn't really matter if two proteins interact if they're never expressed in the same cell. And I think now that we're making this transition to single cell technologies and spatial proteome and transcriptomics, we're going to end up throwing out a lot of the older data sets and going for cell specific data sets. And I think that is going to help us considerably avoid false positive interactions. And then the second thing is I think there's much more need for phenotyping. I do think that high throughput experiments in which, for example, you look at toxicities and you group genes by the fact that they're being upregulated or downregulated in a consistent way in a drug toxicity. I think that those types of networks which are based on shared phenotypes are going to be highly useful. And there should be more experiments along those lines.
Okay. So may I ask, probably a question to you. So you see, you have this Alliance of Genome Resources. Basically, in theory or in practice, you basically have a huge knowledge graph which cover from human to all these low algorithm. So if we can somehow, like in the future, maybe build a huge knowledge graph, link everything together, will help us to create a very nice knowledge graph, especially for currently. There are a lot of deep learning algorithms which they are really powerful by feeding this kind of deep learning algorithm for this type of gene function prediction. Will that be some kind of interesting direction?
Yeah, I think that's, one of the motivations to me to push this forward was something, was trying to do data integration 15 years ago. And the biggest problem was getting all the information into a standard format in one place. So now from, even though the Alliance data so far is not the complete set, it's pretty extensive and you can get it from all the organisms. So I would say it's just sitting there to put into an algorithm, right? It is AI ready,
Okay.
right? It's all ontologized, it's all, it's harmonized, it's ready to go, right? But what's interesting though is if you compare, say, what the IDG and what Tudor's group's doing with and you all are doing with the information in IDG, is it's still it overlaps, but it's not contained, it's not the same set of information. So we need to do is get to the point where we have all the information that's computable and it's all accessible, right? And then everybody can go and do their magics on it, right?
Yeah.
And then figure it out, and then and just drive experiments forward. It's just crazy to waste all this experimental time on these experimentalists trying to, read the literature, right? I mean, just think how much time was wasted.
Yes. Yeah. Okay.
Equal only by the time it took the time wasted to write the papers.
Yeah. So anyway, that's okay. Well, so my view is that I don't really think we should be writing the papers because nobody reads them anyway. So the computer should write the papers, or just not even have it. Okay. So that's the whole point of the structured information, is you have that and then you write a paper.
Yeah.
Anyway.
Okay, so I think we are on time. So thank you. Yeah, it should be a really great talk in the future, probably. Unfortunately, just 1 hour. Okay, I think we have great thanks to both speakers, Paul and Lincoln, and hopefully we have more chance to discuss this. Thank you.
Thank you all.
Bye.
Thanks, everybody. Good to see you. Bye, Glenn.
Bye.
Bye.
Thank you.